Assalamu alaikum everyone. I am Muhammad Hashim Iftikhar, a student of plant breeding and genetics at the University of Agriculture, Faisalabad. Today I'll be presenting on the topic understanding glucosinolates that are nature's protective compounds in crops. So in this presentation we will go through what glucosinolates are, their chemical structure, how they function through hydrolysis, their biological significance, and finally some recent research findings from different crops. So let's start with a general overview. Glucosinolates are sulfur and nitrogen containing secondary metabolites found mainly in the Brassicaceae family, such as mustard, broccoli, and cabbage. They play an important role in plant defense and have several health promoting properties for humans. Now that we know that glucosinolates are, let's look at their chemical structure and composition. Chemically, a glucosinolate molecule consists of three parts: a beta D glucopyranose group, a sulfonated oxime moiety, and a variable side chain derived from amino acids. This side chain determines the type and activity of each glucosinolate, which is why different plants produce different forms. But these compounds become biologically active only when they undergo hydrolysis, which I will explain next. When the plant tissue is damaged, for example by insects or cutting, an enzyme called myrosinase breaks down the glucosinolates. This hydrolysis reaction produces several biologically active products, mainly isothiocyanates, nitriles, and thiocyanates. These products are responsible for the sharp smell and taste of cruciferous vegetables and also for their defensive and medicinal effects. Now let's see why is this reaction so significant both for plants and for us. For plants, these breakdown products act as natural pesticides, protecting them against insects, fungi, and pathogens. For humans, isothiocyanates and related compounds have anticancer, antioxidant, and anti inflammatory properties. They help detoxify the harmful chemicals and strengthen our immune response, making them key contributors to plant based health benefits. After understanding their chemistry and biological roles, let's now move towards some recent studies that explored the glucosinolates in different crops. Here is paper number one. This paper highlights mustard seeds as a rich natural source of both glucosinolates and omega 3 fatty acids. The major glucosinolates identified were sinigrin, which gives mustard its pungent flavor and is very abundant in Brassica gentiana, and cinelbin, a milder compound found in the Sinapis alba. In terms of function, the glucosinolates are converted into isothiocyanates or ITCs, which have multiple pharmacological actions. They are anti-cancer, antimicrobial, antifungal, and anti-inflammatory. For example, they induce apoptosis in the cancer cells, inhibit NF-κB and other phase 1 enzymes, and suppress the harmful microbes such as E. coli and Helicobacter pylori. Clinically, the paper mentioned the use of auriculotherapy with white mustard seeds, which was shown to reduce the body mass index, fatigue, and the anxiety. Moreover, mustard bran was found to lower the postprandial glucose levels, showing potential for managing the metabolic disorders. A second paper focuses on Uh, Lobularia libyca, another member of the Brassicaceae family, the researchers identified three key glucosinolates. Their concentrations were highest in seeds, followed by leaves, and none were detected in the roots. Regarding bioactivity, the extracts showed strong antimicrobial action against Candida albicans and moderate activity against Pseudomonas aeruginosa, but they were inactive against Aspergillus flares. The study also reported cytotoxic effects suggesting a selective anti-cancer potential. The resulting isothiocyanates inhibit protein synthesis and activate the detoxification enzymes, which together contribute to their anti-cancer behavior. Here is a paper 3. The third paper examined interactions among four aliphatic glucosinolates in broccoli. The researchers found that these compounds are mostly antagonistic, means that they can reduce each other's biological potency. In particular, uh, progoitrin was shown to decrease the overall activity of the mixture. However, an optimal balance containing around 80% glucoraphanin produced the strongest anti-cancer activity. The authors emphasized that plant breeding could be used to optimize glucosinolate ratios in broccoli and other crops to enhance their health promoting properties. Here is a case study about broccoli storage effects. It was published in 2022. The treatments used were whole, floret, quarterly, and shredded. These are the findings that with storage, the glucosinolates contents are less. So we can say that overall these studies highlight that glucosinolates are not only essential for the plant defense but also offer remarkable health benefits for the humans. Through biochemical understanding and plant breeding, we can optimize that these compounds to develop more nutritionally valuable and disease resistant crops. Thanks a lot for your kind listenings.